What is the importance of niche versus neutral assembly mechanisms in determining and structuring tropical tree communities? Well, now there is much evidence and studies that shows us that spatial tree distribution is determined by soil and habitat factors such as topography, climate, and hydrology. The clearest evidence for the effect of habitat and soil factors comes from studies conducted at mesoscales and landscape scale. There are several theories that try to test niche assembly due to resource competition. One tells us that tree species in such diverse and rich areas can coexist in a single space of land. The competitive interactions in these rich species areas start acting like a neighborhood with high diversity and high rarity between most of the species, but with a lack of interaction. This lack of interaction suggests that there are competitive differences among species, and such interaction does not determine the structure of the community. But there are major forces that do determine the structure of the community, one being light requirement. Some species would rather grow under high light, and others will choose shade for survival purposes. The other one are pests. Seedlings and saplings of different species have different pest resistance. And the third one being positioning, due to inferior species being better colonizers than superior species. But how does all these theories and evidence about soil translate into niche assembly at a local scale? Does this affect areas that are less than 1 square kilometer? And are there easily visible differences between the spatial distribution of trees in three forest plots of the neotropics? For such answer, this research studied the forest at Barro Colorado Island in Panama, La Planada, Colombia, and Yasuni National Park in Ecuador. There lies the importance of this research. Instead of focusing on small fractions of the tree species in any community or related communities, it made a community wide analysis and a high resolution spatial mapping of soil resources. As for the methodology to do so, research was conducted at three main sites. In Barro Colorado Island, a 50-hectare plot was determined to represent the lowland semi-deciduous moist forest. In Yasuni Park, a 50-hectare plot was determined to represent the lowland evergreen forest. And at La Planada, a 25-hectare plot was determined to represent the evergreen montane forest. At each site, at least 300 samples were taken. Each sample contained 50 grams of topsoil, 20 of which were used to test the moisture content. The remaining grams were used to make elemental analysis to extract soil cations such as nitrogen, phosphorus, calcium, magnesium, and mineral nitrogen. As for the results of this methodology, similar effects of species distributions were seen at Barro Colorado Island and La Planada. At all sites, nitrogen and phosphorus were no stronger than calcium, magnesium, and iron as a determining factor of species distribution. Aluminium is comparable with major nutrients in the effects of species distribution, and as for an overall analysis, there was a non-uniform patchy distributions of nutrient in the soil. As for some more general results, Barro Colorado Island had a leading species distribution based on boron and potassium. Yasuni had a leading distribution based on calcium and magnesium, while La Planada had a leading species distribution based on potassium, phosphorus, iron, and nitrogen. As for the conclusion based on previous studies, these results show us a disruption from the idea that phosphorus was more limiting than nitrogen in mature lowland tropical forests, like the one seen in Barro Colorado Island and Yasuni, and it also disrupts the idea that nitrogen is more limiting in montane forests, like the one seen in La Planada. In La Planada, there doesn't seem to be a general pattern followed, since nitrogen and phosphorus have the same effect, while in Barro Colorado Island, nitrogen and mineral nitrogen have a bigger effect, and in Yasuni, phosphorus has a bigger effect.